Welcome in this new video. In this video we are going to study about applications of biotechnology, that how pest resistant plants are formed and what is RNA interference and how RNA interference works. If you want to know all the answers of these questions, you have to watch this video completely. So several nematode parasites affect wide variety of plants and animals. So for pest resistant plants, RNA interference is used. Meloidegyne incognita is a parasite which affects the roots of tobacco plant. Due to the infection of this pathogen, production of tobacco plants decreases. Here you can see that in tobacco roots, infections occur due to this pathogen. Now we are using RNA interference. Some points related with RNA interference we have to be remembered because this can be asked in exams too, which is RNA interference is a natural occurring mechanism, that is, it is already exist in the nature. We just use this RNA interference for our purpose too. So it is natural occurring mechanism and it is present in all eukaryotic cells as a cellular defense. That means RNA interference is present in every eukaryotic cell, maybe in plant cell, animal cell or other animal or organism cells. Okay, in every eukaryotic cells RNA interference is present. It protects the cell from viruses too. Now we have discussed properties of RNA interference. Now mechanism of RNA interference we are going to study. To understand the mechanism of RNA interference, we have to understand the mechanism of infection of the pathogen. That means many pathogens produce many types of proteins or enzymes which affect the host organism, and some pathogens also introduce a RNA or DNA into the host so that they can affect the host cell from inside. That means they are endoparasite. Now according to central dogma theory, DNA forms RNA and RNA forms protein. So this protein is the main cause of pathogen. But if we stop the translation of the RNA to protein, then we can prevent the infection by the pathogen. So we have to silence this mRNA so that this do not produce the protein. So protein will not be formed. So we can understand this from a diagram which you can see in the right side. Here it is a host cell. Here this is a mRNA which is pathogenic, which cause infection in the cell. Now a double-stranded RNA or say complementary RNA is present or introduced inside the cell. This double-stranded RNA silence the mRNA. So this is the technique of RNA interference, where a double-stranded RNA which contain both sense and antisense of the mRNA molecule. So this silence the mRNA molecule, original mRNA molecule, which is responsible for the infection. Now how this double-stranded RNA is introduced in the host cell? Because in eukaryotic cells double-stranded RNA is not present, so we have to be introduced into the host cell. So here biotechnology works, where we introduce the double-stranded RNA into the host cell with the help of transposomes, which are mobile genetic elements which introduce the foreign genetic material into the host. And the second, which is most important in plant biotechnology, that is agrobacterium vector. This is a vector which can directly introduce or inject a foreign DNA into the plants. If we talk about animals, we have retroviruses for this purpose, and in plants agrobacterium vector helps to introduce this DNA. So here agrobacterium introduce a DNA which produce two types of RNA, that means sense RNA or antisense RNA. Due to the complementary of these both antisense and sense RNA, they form double-stranded RNA, and this double-stranded RNA is responsible for the silencing of the mRNA. So here you can see that double-stranded RNA is responsible for the silencing of the mRNA, and due to this, pathogen do not cause any infection into the host or in the plant. With the help of this, we can increase the production of the crops. Now, so again I am summarizing all these points in this video which are related with RNA interference. That means RNA interference is a natural occurring mechanism. It is present in all eukaryotic cells. RNA interference means double-stranded RNA silence the mRNA which is introduced by the pathogen, and double-stranded RNA is introduced by the agrobacterium vector which introduce DNA and that contain both sense and antisense RNA, and that form double-stranded RNA. So this is the complete summary of this video, RNA interference. So if you understand this RNA interference, subscribe to my channel will be incredible. Thanks for watching. See you in the next video.